Hello everyone, and thank you for attending today's webinar, How to Achieve Sensitive and Specific Oligonucleotide Quantification by Hybrid Immunoassay and LC-MS/MS. I'm Tristan Free, Assistant Editor at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speakers, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around and get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right hand corner. Please let us know your thoughts on today's webinar by tweeting at us at BioAnalysisZone using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. And an on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we are delighted to have Chenyi Pan, Group Leader of Biologic Services, and Jiling Zhang, Lab Member of Bioanalytical Services at Frontage Laboratories. Dr. Chenyi Pan received his BS from Zhejiang University, China, and his PhD in Biology from the Georgia Institute of Technology, GA, USA. His PhD research was focused on the epigenetic regulation of stem cells in cancer. Before joining Frontage in 2017, Dr. Pan worked as an NSF EBIX postdoctoral fellow in biomedical engineering at Georgia Tech, where he developed novel biosynthesis systems to monitor intracellular redox status during stem cell differentiation. At Frontage, Dr. Pan is responsible for immunoassay development and validation, as well as communication with clients on deliverables. He has strong expertise in PK, immunogenicity, and biomarker assays based on various platforms, including ELISA, MSD electrochemiluminescence, Quanteric Simoa, and ELA. Working in a GLP and CLIA regulated environment, he has been helping biopharmaceutical companies with preclinical and clinical drug development in fields such as immuno-oncology, neurology, and diabetes. Dr. Jiling Zhang received his BS and MS in Tsinghua University, China, and his PhD in biomedical engineering from Drexel University. His PhD research was focused on the development of drug delivery systems for therapeutic proteins and small molecular drugs. After joining Frontage in 2016, Dr. Zhang continues to use LC-MS platforms to analyze large molecules, including antibody-drug conjugates, oligonucleotides, proteins, and polysaccharide biomarkers. He has demonstrated strong expertise in affinity capture, protein structure, and LC-MS techniques. Under a GLP-regulated environment, Dr. Zhang also works as principal investigator for method development, validation, and sample analysis, supporting preclinical and clinical studies for pharmaceutical companies. We invite the audience to submit any questions you may have for our presenter throughout the webinar using the Q&A widget to the right-hand side of your screen. These will be followed up offline after the webinar has finished. Thank you again everyone for joining us today. I'll now hand over to Chenyi Pan and Jiling Zhang to start today's presentation.
Thank you, Tristy, for having us here, and a very nice introduction. And today's webinar will focus on highly sensitive and specific quantification of oligonucleotides in biologic matrices by hybridization immunoassay and LC-MS/MS methods. I'm Chenyi Pan.
I'm Jiling Zhang.
Today we will focus on the analytical challenges and methods of oligonucleotide or OGN therapeutics. First, we'll give you an overview of OGN-based therapeutics, which include the mechanism of the action, considerations for OGN bioanalysis, pros and cons of existing bioanalytical methods, and the challenges we are confronted. Then we'll focus on the discussion of our approaches to tackle these challenges by presenting 2 case studies for OGN bioanalysis based on ligand binding assay and LC-MS platforms. OGN therapeutics have emerged as an important therapeutic modality for disease treatment in recent years. There have been several approved OGN drugs, including nusinersen from Biogen and patisiran from Alnylam, and there's a broad pipeline in development from various companies. Advances in our understanding of human disease and the availability of the full human genome sequence have created numerous therapeutic applications for these drugs. This therapeutic modality consists of a diverse class of drugs such as antisense oligonucleotides, small interfering RNAs, microRNAs, aptamers, etc. Typically, OGN drugs that are designed for modulation of protein biosynthesis function through 3 different mechanisms. ASOs can be designed to cause exon skipping or inclusion by simply binding to the pre-mRNA. They can also be designed to promote RNA degradation by recruiting RNase H. Double-stranded RNAs are able to suppress gene expression through RNA interference that we are more familiar with. Given their unique features and mechanism of action, there are specific considerations for bioanalysis of OGN therapeutics. In general, they are designed to function inside the cells and therefore present unique challenges for drug design and PK/PD studies. Chemical modifications can be added to promote target interaction, stability, and efficacy, and also lead to PD/PK advantage. They are usually degraded by nucleases instead of cytochrome P450 enzymes, and therefore cytochrome-mediated effects on OGN therapeutics are predicted to be minimal. To support the preclinical and clinical PK studies of OGN drugs, it is essential to develop a simple, specific, and sensitive method for an accurate quantification in biological matrices. Studies have shown that plasma concentrations of antisense OGNs and posted distribution phase correlate very well with the target tissue concentrations and can serve as surrogate measure of tissue concentrations. Today, many different bioanalytical methods have been developed for OGN quantification. Each of them has its own pros and cons. Particularly, RT-qPCR offers unmatched sensitivity and a broad dynamic range of quantification, but it lacks specificity and needs specific reagents. Existing hybridization-based ELISA has high sensitivity and requires little sample preparation, while it also has some issues like low specificity, relatively narrow dynamic range, and lack of metabolite information.
In addition to ligand binding methods, chromatography methods can also be used for the analysis of oligonucleotides. Chromatography methods provide better specificity, especially when mass spec is coupled. It can also be used to obtain metabolite information. However, the sensitivity is an issue, and intensive sample cleanup is needed for these methods.
The issues existing in different bioanalytical methods represent the current challenges in OGN quantification. Several major bioanalytical methods like qPCR and hybridization ELISA lack selectivity and specificity. In some cases, an ultra-high sensitivity is required for OGN quantification in plasma or serum. The large size and negative charge of the OGN molecules can raise challenges in sensitivity for the LC-MS method. OGN molecules may be unstable in vivo due to the nucleus activities. Also, some bioanalytical methods require heavy sample preparation. To tackle these challenges, we have developed multiple approaches to meet the specific needs. These approaches include ligand-binding methods such as MST electrochemiluminescence and ELISA-based assays, and chromatographic methods. Our ligand-binding methods can achieve an unparalleled sensitivity and meet the most stringent requirement. And these assays are particularly powerful in quantifying OGN drugs in plasma and serum where low levels are expected.
Chromatography methods have different applications. LC-mass method shows excellent specificity and is good for tissue sample analysis, as the tissue sample usually show higher concentration of OGN. Both LC-mass and LC-UV can be used for the metabolite quantitation. Furthermore, both non-modified and chemically modified OGN can be analyzed by LC-mass method.
In the next part, we'll present two case studies for ASO quantification using these methods. ASOs are short, chemically modified, single-stranded DNA or RNA nucleotides that specifically target genes of interest and regularly target protein biosynthesis. Typical hybridization ELISA and LC-MS methods have shown limited sensitivity in quantifying these agents. In this first case study, we have developed and validated MSD-ECL and ELISA methods for ultra-sensitive ASO quantification in human plasma. Method validation parameters include the calibration range and QC samples, precision and accuracy, selectivity, dilutional linearity, prozone, and specificity. The model ASO analyte we used here is a 20 nucleotide DNA fragment. Unlike a standard ELISA, which uses a specific antibody or recombinant target protein, the hybridization ELISA utilizes a complementary single-stranded oligonucleotide capture probe. The capture probe here is a 3' and biotinylated 29-mer DNA fragment, which was designed with the first 20-mer sequence from the 3' and complementary to the analyte. The detection probes are 9-mer DNA oligonucleotides complementary to the 5' and overhang of the capture probe, with 5' and phosphorylated and 3' and deoxygenin or ruthenium labeled. The assay procedure was adapted from a previously published paper with some modifications. Briefly, plasma samples containing the analyte were incubated with the capture probe for hybridization and then loaded onto an avidin-coated acid plate for immobilization. Afterwards, T4 DNA ligase and the detection probe was added to the plate for ligation of the detection probe to the analyte. On the second day, S1 nucleus was used to truncate the probes that were non-ligated or non-specifically hybridized. Then the plate was read on an MSD image sector or a fluorescent plate reader. There are some critical steps involved in these assays. The range of quantification can be modulated by using different concentrations of capture and detection probes. The use of S1 nucleus will dramatically increase the assay sensitivity. For the MSD-ECL method, the ruthenium-labeled probe was read on MSD image sector, and for the ELISA method, the deoxygenin-labeled probe was quantitated by an alkaline phosphatase conjugated antibody, and the ethyl for fluorescent substrate system. We have validated these assays for a series of parameters, including the calibration range, precision and accuracy, selectivity, linearity of dilution, and specificity. The data generated from these tests will be presented in the next several slides. In this slide, it shows that both methods achieved an ultra-high sensitivity with an LOD of 2 picomolar. That is about 12 picogram per ml. This is among the highest sensitivity that has ever been reported by similar studies. There was a linear signal increase within the calibration range from 4 picomolar to 1,024 picomolar. As shown by these representative calibration curves, both MSD-ECL and ELISA methods showed very good CV and RE for all the standards. Both methods achieved high precision and accuracy for the standards. We tested 5 QC levels spanning from the LOQ to the UOQ for inter-run precision and accuracy, and all QC levels met the 20% CV and RE criteria for both methods. To test the selectivity of these methods in presence of different matrix components, we analyzed the 10 individual plasma samples at the LOQ level. All the samples met the 20% CV and RE criteria. Next, we tested the analyte dilutability. The ASO was diluted starting from the reference stock to a level below the LOQ by a 4-fold zero dilution. As you can see in this table, both assays showed excellent dilutional linearity, especially for MSD-ECL method. The analyte can be diluted for more than 80 million times. So we believe that transformation of ELISA onto the MSD-ECL platform by directly labeling the detection probe with ruthenium improved the assay performance. Given the better assay perform' of the MSD-ECL method, we tested it for specificity on train shortened metabolites. These metabolites include 3' and shortened ones, such as 3' and N-1, -2, and -3, and the 5' and shortened metabolites, N-1. These metabolites were spiked into plasma at different QC levels and measured against the analyte calibration curve. As depicted in this slide, our method didn't pick up any of the 3' and shortened metabolites for all the QC levels, suggesting that it can distinguish the analyte very well from this group of metabolites. Unfortunately, like other similar methods, our method wasn't able to distinguish the 5' and short N-1 metabolite from its parent. The cross-reactivity ranges from 80 to 95%. However, since the majority of the metabolites for ASO drugs existing in vivo are 3' and shortened metabolites, the hybridization ELISA method still can serve our purpose of ASO bioanalysis in biological matrices, where a high sensitivity is required. This issue might be overcome by optimizing the probe design. And now, we would like to switch gears and move on to the second case study, which is the bioanalysis of OGN using LC-MS-based assays. Now, I will hand over the microphone to my colleague, Dr. Zhiling Zhang.
LC-MS-based assay is a very powerful tool for OGN and its metabolites. Although sensitivity can be an issue, especially for plasma samples, it's still good for tissue samples while the oligonucleotide concentration is relatively higher. In addition to traditional OGN, chemically modified oligonucleotides can also be quantitated. This type of assays can also be used for identification and quantification of OGN metabolites. This slide shows a typical procedure to analyze oligonucleotides in tissue samples. After homogenization, tissue samples will be double extracted. Liquid extraction followed by solid phase extraction. In the LC-mass setting, ion pairing reagents are used to improve the retention and separation of analytes. mRNA modes are used so that the multiple molecules can be monitored simultaneously. For example, one ASO and multiple ASO metabolites can be quantitated from single sample injection. Ion pairing chromatography results are shown here. We can see without ion pairing reagent, the OGM will not be retained, and all compounds are eluted together very early. And when we use the ion pairing reagent, multiple compounds can be well separated, and then they can be quantitated individually. It's very common that multiple tissues are collected and require the analysis while sample number of each type is very limited. An alternative approach can be applied while a surrogate matrix or one of the target matrix will be used to prepare the calibration standard, with QC being prepared in each type of study matrix for accuracy and precision check. In some projects, we analyze the same oligonucleotides in 15 tissue types, and the results are good. Here, we just show the QC accuracy and precision from 4 different tissue types. We can see the QC BIOS and CV are all within the acceptance criteria. Plasma samples can be analyzed for oligonucleotides as well by SP extraction followed by injection into LC-mass. The sensitivity is around 10 nanomolar, which may be an issue for oligonucleotide assay in plasma samples. Chromatographic method can also be used for OGM metabolite analysis. This slide shows a chromatogram of this analysis. Even if the OGM and its metabolites were not completely separated by the chromatography, they were monitored using different mass transitions, so that each compound can be quantitated separately. Oligonucleotide and its metabolite can also be separated by LC with longer elution time, and then characterized with UV detector. In this case, the metabolite can also be quantitated even without reference standard. In addition to conventional oligonucleotides, we also analyze chemically modified oligonucleotides, which can be extracted by solid phase extraction and then injected into LC-mass for analysis. Due to the chemical nature of modification, the performance of these chemically modified oligonucleotides in LC-mass can be quite different. For example, if the backbone of oligonucleotides was modified in phosphate group, the charge of the analyte could be altered. In this case, reverse the phase LC without using ion pairing reagent can be used to detect the OGM, and the sensitivity can be improved from 10 nanomolar to 1 nanomolar. Here are our conclusions. Our hybridization immunoassays achieved an ultra-high sensitivity for ASO quantification. The performance of our methods passed the most stringent criteria in all the validation parameters tested. A high level of specificity to a series of potential 3-ended shortened metabolite was achieved on the MSD ECL assay platform. LC-mass method provides very good specificity and has demonstrated a great assay performance for the analysis of ASO in tissue samples. It provides sufficient sensitivity for modified oligonucleotides, and it's a great tool to identify and quantitate oligonucleotide metabolites. Thank you again for your time. We are Dr. Ziling Zhang and Chengyi Pan from Frontage Lab.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value. If you have any other questions for Chengyi Pan and Ziling Zhang, please feel free to send them in via email to webinars@bioanalysis-zone.com. I would like to once again thank our wonderful presenters, Chengyi Pan and Ziling Zhang, as well as you, our listeners, for your time. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at bioanalysis-zone. Don't forget to visit us at www.bioanalysis-zone.com for more webinars. Thank you again for attending, and goodbye.